So today we're actually talking about 3 different exercises. We're talking about the fluothioglycolate media that we commonly abbreviate FTM. And these you inoculated last week. These were the tubes that I had boiling, and then I had to wait for them to cool down. They were the ones that had the little pink area at the top. And some of you guys don't actually have these tubes in your rack right now because I had to refrigerate them. And you'll find out today in the lecture why I needed to do that. Anaerobic jar, you guys read and should have taken notes on for homework. We won't actually do this experiment. We don't have enough of these jars working properly for all the classes to be able to do this experiment. So we do it as an in-class discussion, so we're going to discuss it now. You guys will still be quizzed and tested on it, even though we're not going to perform the actual experiment. Page 6 in your lab manual is we're going to refer to today because it's going to talk about the importance of controls, right? And as we do a lot of these tests, we're going to be doing controls. So one of the things that we did last time too is we even set up a control right on our gram stain slide, right? Of gram positive versus gram negative to be able to compare to our unknown. And so today, after I lecture, after we record our results for FTM and then also motility test, then you guys will create some more smears for staining, and you also stain your gram slides that we made last week. So fluothioglycolate media, as I said, this was the one that ended up with a little bit of pink here at the top, and there's a reason for that. This has an indicator in it. So the reason why we use this media, it has sodium thioglycolate, obviously in the name, and L-cytosine in the media. From your homework, what does this do? What do these 2 components do? You said it. Reduces oxygen, right? So we're actually creating an oxygen-free environment in the bottom of this tube, right? So we're removing free oxygen from the media. There was a little bit of agar in our tubes too, right? So if you had a lot of experience with inoculating broth cultures as versus this culture, you may have felt a little bit of a difference. But this is also why when I told you guys to inoculate, that you had to go all the way down and inoculate up through the media. The problem with that is I showed that like 2 or 3 times in the one that I was inoculating. So you guys were like shaking like crazy and probably introduced more oxygen into your tubes than need be. So we'll talk about that when we look at our results a little bit later. But again, this is meant to slow the movement, right? Because these are not sealed tubes. So oxygen does diffuse in over time. And so that's one of the main reasons why some of them, I took them out of the incubator and put them in the refrigerator, was to slow their growth. Because the oxygen was going to continue to diffuse in. And so therefore, you would end up with growth all throughout this tube if I didn't stop this process, right? And you'll see that as we look at some of these tubes today, at your tubes, that you may even see the oxygen has diffused quite far down in the tubes. Now, of course, I'm saying oxygen, right? Can anybody see oxygen? No, you can't see it. So that's why you have a pink layer here. There's an indicator, right, called resurin that shows you where the oxygen is, because it reacts with the oxygen and it produces this pink color, right? So it allows us to see where the oxygen is in this tube. So is it oxidized or reduced in the presence of oxygen? So there's an old saying that I always remember, right? Oil rig. Oxidation is the loss of electrons. Reduction is the gain of electrons. So in this case, this reagent becomes oxidized, right? It actually loses, right? Oxidation is the loss, actually loses electrons in the presence of oxygen. All right, and it's going to give us this pink color. So why is there a colored band? Well, because the indicator is there, right? And allows us to see where the oxygen is. Which is more desirable? Do we want a thick layer or a thin layer, do you think? We want a thin layer. We want just a little layer of oxygen. So any of our organisms that need oxygen have this space to grow. If you have a lot of oxygen in this tube, then you don't have any space for your anaerobes to grow. And that's the main reason why we're using this media. You'll notice our question for our unknown report is, can your organism grow anaerobically, right? We want to know, can it grow without oxygen at the bottom of this tube? Right, so resorrin is that dye that is pink when it's oxidized, and it's colorless when it's reduced, right? So it's all throughout the media, right, but it's not producing a color towards the bottom of our tube. So hopefully we just see it at the top, right? The whole purpose of using this media is to create an anaerobic environment, right, in the bottom of that tube. So these are the different growth patterns you can see based on the different oxygen tolerances and needs, requirements for the different types of organisms that are out there. So a strict aerobe, right, needs oxygen to grow, will only grow in the presence of oxygen. So where would it grow in our tubes? At the top, only where the oxygen is, right? So in this case, which tube do you see growth only at the top? E, you see it at the top, and D, right? But which one do you see the most growth? D, right? And that is our strict aerobe. And some of you guys have strict aerobes as your unknown organism. And because of that, I had to refrigerate it, right? Because the incubation period would have been too long, right? You would have to come in and check it the next day for it to be the most accurate, because of that diffusion of oxygen that we can't stop. Our strict anaerobe, so this means completely without oxygen, right? So which one is growing where there isn't oxygen? Where do you see turbidity or growth? Which one?
A.
A is growing at the top too. It's growing at the bottom too, right? C, do you see how the pink zone doesn't have any growth in it? That's the oxygen zone. And then this is the rest of the zone that's the non-oxygen. Do you see how there is growth there? This is our strict anaerobe, excuse me. These guys are actually harmed by oxygen and cannot grow in the presence of oxygen. They also aren't utilizing it obviously for their metabolism, otherwise they'd be dead. Right? We don't have any of these in this class. It's too hard to create a strict anaerobic environment for our organisms, right? So none of you guys are going to have this situation. Microaerophiles. Micro implies what? Small, right? So these guys need a small amount of oxygen, right? Not a large amount of oxygen. So where do you see them growing where there might be just a little bit of oxygen in the tube? This is the E, right? So notice, you see that little bit of turbidity right here? Right below the oxygen zone, right? So right at the bottom where there's the least amount of oxygen, but not completely devoid of oxygen. There's no growth at the bottom, right? Or the rest of the tube. So our tube E, close to the top, just below the oxygen zone, is where these guys are going to grow. Now, facultative anaerobe. Anaerobe implies that it can grow anaerobically, right? So we'll expect to see growth where there isn't any oxygen. Two tubes are like that, A and B, right? But there's a difference between A and B, right? What do you notice about the growth in A? It's pretty uniform all throughout the tube, right? Really no difference between our oxygen and non-oxygen zone. Where for B, do you see how there's more growth here and then less growth in the non-oxygen zone, in the anaerobic zone? This is our facultative anaerobe. It can grow anaerobically, as you can see, but facultative refers to the ability to change. And these guys can grow better in the presence of oxygen. Hence why you see more growth in the oxygen zone. a lot of you guys have facultative anaerobes, right? So if you look at your FTM tubes, which you might even have in front of you right now, you might notice growth all throughout, but if you really look, you may have a concentration of growth at the top of that tube. But then again, these tubes have gone for a long time, so oxygen is probably diffused throughout the entire tube, right? So you might not see that difference in zones. Aerotolerant anaerobes, anaerobe referring to the fact that it doesn't need oxygen to grow, aerotolerant referring to that it can tolerate oxygen, not that it's utilizing it. So of course the only tube we're left with is A, right? And that's where you have nice even growth all throughout because oxygen is not harming it like you see with our anaerobes, but they're also not utilizing it, so you don't see this difference between the oxygen and non-oxygen zone. We may or may not have aerotolerant organisms, right? Most of you guys have facultative, right? So why do we see differences in growth? It has to do with, as I said, what they're using for energy. So the catabolic process, when they're breaking down large molecules into smaller molecules to generate energy, right? And then that energy is later consumed in a process of anabolism where they're actually putting molecules together to build cells, right? When they grow and multiply, right? So it's this cycling of energy from one molecular form to another. Carbohydrates are the main source, right? What we refer to as sugars. Glucose being the most predominant one utilized by most catabolic systems in our cells as well as these bacterial cells. So it can go through a process where it's split up, right? That process is referred to as glycolysis, if you remember from your biology classes. And then the Krebs cycle or the TCA or citric acid cycle, right, where they further break down the products from glycolysis and generate more energy. And then some of those products take electrons from the Krebs cycle and from glycolysis and bring them to the electron transport chain, right, where they generate even more energy. The most common energy carriers, do you guys remember what usually carries the energy from one place to another? It's an abbreviation. ATP, right, or GTP, sometimes you'll see, right? So it's that phosphate bond, right, that's what the P stands for, that holds that energy and brings it from one place to another. So in glycolysis, a small amount of ATP is produced. It's at what we call the substrate level, at the enzymatic reactions that are happening. Most of it is going to come from electron carriers that are going to go to our electron transport chain. And in that case, it's referred to as oxidative phosphorylation. And this, again, has to do with electrons, right? When we say oxidize, we're not referring to oxygen. We're referring to electrons, right? The movement of electrons, in this case, the loss of electrons, right, and being donated. And so that's a large percentage of the energy that's produced. The problem is those electrons have to go somewhere, right? And the final electron acceptor for aerobic respiration is oxygen. But some organisms cannot utilize the electron transport chain because oxygen may not be available, right? They might be in an anaerobic environment. In that case, they're going to use glycolysis, right? And that's it. They can't utilize the Krebs cycle or the electron transport chain because they're not using the electron transport chain because they don't have anything to give the electrons to, right? There's no oxygen available. So they can't do the Krebs cycle. It generates too many electron carriers that don't have any place to drop off their electrons. So then they're stuck with glycolysis, and they still have a problem because there's some electron carriers used in glycolysis, just not as many as the Krebs cycle. The way they regenerate those is they drop the electrons off at some of the products of fermentation. So they add it back to the breakdown products that they've generated in glycolysis. And so this is how we get a lot of the alcohols that are produced. sometimes also the gases, and the acids, the different acids that are produced during fermentation. So fermentation is an anaerobic process, right? It's not utilizing oxygen. And so when we start studying, and as you guys start reading for the next one, a lot of these processes that we look at are going to be fermentation processes, right? Where we're going to look at these production of these products like alcohols and gases and acids, right? So keep those in mind as we go through. So organisms use all three pathways to catabolize, right? Break down sugar. Are said to what we call is respire, right? If they're doing glycolysis, Krebs cycle, electron transportation. This is referred to as respiration. This is different from what, some people refer to as breathing, right? Moving oxygen and gases in and out of our bodies. Respiration is the actual chemical process of breaking down sugars, right? And generating that energy. And where do the electrons go? If it's aerobic respiration, they're going to oxygen, and that's why you need oxygen in order to respire, right? In order for that metabolic process to happen. Now, of course, right, in that process, we actually form what's called metabolic water, because the electrons are donated to oxygen and protons are also added, and you actually form water. For anaerobic respiration, anaerobic basically means that oxygen is not your final electron acceptor. They're using something else to accept those electrons. So lots of inorganic molecules are utilized by organisms. So these are just a couple of examples, right, of some of the molecules that organisms use. So sulfate, they could add it to make sulfite, and then they can even further add electrons to it, oxidize it to form hydrogen sulfide. Others can reduce nitrate to nitrite and then to ammonia, right? And these are common nitrogen sources. And these bacteria are very important in our soil, right, for helping recycle nitrogen through the system. Others actually remove nitrogen from the systems because they go from nitrite to nitrite to nitric oxide to nitrite I hate these chemical names, to nitrogen gas, right? So they actually remove nitrogen from the soil. Those guys aren't the good ones. We want the ones that go the other way, that bring nitrogen into the soil from the atmosphere. So if organisms are grown on a media that contains sugar but lacks compounds necessary for anaerobic respiration, then they have two options, right? They can grow well, i.e. quickly via aerobic respiration, if they can do that, right? Or they'll grow poorly, i.e. slowly, using fermentation, right? And that's definitely what we see with our facultative anaerobes, right? They're doing fermentation when there isn't any oxygen. So in reality, not all organisms have that ability to metabolize in different ways. Our facultative anaerobes are one of the examples of where they have choices. But many don't have these alternatives that we talked about. So on our planet, actually, most organisms probably to begin with were anaerobes. And it wasn't until photosynthesis started taking place, which actually produces excess oxygen, that you could actually evolve into being an aerobe, right? Or a facultative anaerobe where you can change and utilize. So, and in fact, when we think photosynthesis, you guys think plants most of the time, don't you? Right? But it actually is the cyanobacteria and our algae and our oceans that produce most of the excess oxygen. Plants actually utilize oxygen just about as much as they produce. So they're not the ones contributing to the oxygen in our atmosphere. So this diagram is just a dot plot diagram, just representing growth by dots. And again, this is to represent our FTM tube. So in the top, right? If it's properly prepared and our media is fresh, then we just have a little zone here at the top of oxygen and then no oxygen at the bottom of the tubes, right? So our obligate aerobes would have growth only in that oxygen zone, right? So number 1 represents our obligate aerobes. Number 4, right, is our microaerophiles, because again, they're going to be right below that oxygen zone, right? They're not going to be completely at the top. They're going to be where there's a little less oxygen. Which one would be our obligate anaerobes? Definitely 2, right? Notice growth is only down in the anaerobic environment, the bottom of the tube. Which one is going to be our aerotolerant anaerobes? Only got 2 choices left. 3 or 5, right? What's the difference between these 2 tubes? 3 has more dots here at the top, right? That's in the oxygen zone. Aerotolerant implies that they can tolerate oxygen. Do they utilize it though? No. So their growth pattern shouldn't change in the oxygen zone, right? So they're number 5. Their growth pattern is uniform all throughout the tube. In the oxygen and non-oxygen zone, we have about the same amount of growth, right? Same concentration of dots. So by default, 3 is our facultative anaerobe because you see that greater growth in the aerobic zone, right? Because these guys can generate more energy, right? Because they're going to use glycolysis, Krebs cycle, and electron transport chain. Where down here, they're doing fermentation, right? And they're not generating as much energy in that anaerobic zone. So why is it important that the media be fresh? What happens over time? That oxygen is going to diffuse down, right? So which type of organism would most likely be negatively affected by old media having a lot of oxygen in it? The anaerobes, right? So our anaerobe would have very little space to grow, right? Our microaerophiles, not so much, right? Because again, they do need a little bit of oxygen. There might be enough area. Our facultative anaerobes would be okay, but again, we wouldn't really be able to see that they're growing anaerobically if we don't have a lot. So who would be really happy about all this extra oxygen, right? Our aerobes, right? Our aerobes would be the most happy about that situation. So I've been saying this, right? But let's review and put it all together. Which ones require oxygen for their metabolism? Okay? So our obligate strict aerobes, as their name implies, absolutely yes, right? Our obligate or strict, these 2 terms are used interchangeably. Anaerobes, do they require oxygen? Definitely not. Microaerophiles, yes, but small concentrations of it, right? Facultative anaerobes, do they require it? Do they have to have it? No, right? As their name implies, they can grow anaerobically. Aerotolerant anaerobes, do they require oxygen? No, they don't use it at all. Growth in the presence of oxygen. So obviously aerobes require it, have to have it, so they're obviously going to grow in the presence of it. Anaerobes, can they grow in the presence of oxygen? No, they're actually harmed by it. Microaerophiles, yes, but again, small concentrations of it. Facultative anaerobes, can they grow in the presence of oxygen? Yes, quite abundantly at that. Aerotolerant anaerobes, yes, as the name implies, aerotolerant. They can tolerate oxygen. They can grow in the presence of it. Growth without oxygen. Can our aerobes grow without oxygen? Absolutely no. Obligate strict anaerobes. Yes, it's actually the environment they prefer. Microaerophiles, can they grow without any oxygen? No, they're not going to grow. Facultative anaerobes. They grow? Is their growth affected by the lack of oxygen? Yes, they're going to grow less, right? They're going to grow modestly. They're going to grow less than the abundant conditions of when they have oxygen. Aerotolerant anaerobes, can they grow without oxygen? Yes, as their name implies, they grow anaerobically, right? Not affected by oxygen. So our scientific method for FTM tubes, right? What's our hypotheses? What experiment are we doing, right? What's the method that we're going to employ? Proper method, right? What are the outcomes? What should I record? How should I report them? And what does this data mean, right? So what is the oxygen requirement for our particular organism, right, is our main question we're asking. Can it grow anaerobically instead of or in addition to aerobically? So how are we testing this? A loop of bacteria is gently swirled, right? As I said, we kind of probably swirled too much, introduced a little too much oxygen into our tubes. The culture tube, the FTM is an undefined rich media which contains glucose and as a carbon source, which can be used to break down aerobically, or it can be fermented anaerobically. So the reducing agents, as we said, remove the oxygen, right? The resorption lets us know where the oxygen actually is in these tubes, right? And the agar helps slow the diffusion, again, of the oxygen into those tubes. So what could we see? Well, it depends on what your organism is, right? As we've already discussed, you're going to see these different growth patterns based on their different abilities in our tubes. What does this data mean? Right? Oxygen utilization is indication of energy metabolism. Either bacteria respire aerobically and you get lots of energy, or they're going to ferment, right? Or grow anaerobically and they're going to use less energy. Because remember, as I said, we don't have any strict aerobes in this class, right? Because it's too hard for us to maintain that environment, right? Your tubes right now on your lab bench, your organisms would be dying, right? Because this is definitely an aerobic environment. Okay. So anaerobic jar, again, we're dealing and we're trying to create an anaerobic environment. So we have to have some way to indicate when we have that anaerobic environment, when there isn't any oxygen, but we can't see gases with our eyes, right? So we need some type of indicator. The indicator that we use for this test is different than the resorption. We use methylene blue. Typically it's in a strip or in a tablet. Were you going to grab it, Peter?
No, I'm actually looking for tweezers.
Tweezers, they're up front. Yeah. So we've used methylene blue dye, right? And again, our room is under aerobic conditions. So it's blue. But under anaerobic conditions, it is, in fact, colorless. So of course, again, this room is aerobic. So what color is it? Blue. So you'll notice in this jar I have at the front of the room, you see the tablet associated with that packet that's in there, it's blue, right? That's because that's aerobic right now. This is a picture of when we did set up this experiment, right? And you can clearly see that tablet is colorless, right? There's no blue color to it. So we know by looking at that indicator that this jar at this particular time frame when I took the picture was under anaerobic conditions. Right? There wasn't any oxygen in this jar.
Can I interject?
Yeah?
To help you remember this, do you recall using methylene blue before in the class? Yeah, it's one of the simple stains that we use. So it's blue when we do our staining procedure with the air in the lab.
Yeah.
We also put it in the place that we used last week for counting colonies that was eosine methylene blue.
Mm-hmm.
So again, it's blue in the air. It's blue in color is in the air.
Yeah. So think about the environment that we're mostly in, aerobic, it's blue. So note that it's white or colorless. And so what type of environment? anaerobic. This one, again, notice it's blue, right? And that's because this one was left out, okay, on the bench top, and so therefore it was exposed to oxygen and it turned blue. So, aerobic environment. So what happens? So what that packet is, that packet that we're looking at is a gas pack generator. And it comes sealed, right? And we open it up and then we put it into the jar and seal up the jar. What happens is hydrogen gas and carbon dioxide gas form. But the hydrogen gas is what binds to the oxygen and what molecule it forms. What do you get when you get hydrogen and oxygen? Water, right? You get water. So another picture you'll see, right? You see the condensation inside? That's the formation of that water as the oxygen is being consumed by the combination of the hydrogen and oxygen gas. So that's another good indicator, right, that things are working properly as they should. So if the environment is anaerobic inside the jar, how will it affect the growth of these organisms? Well, we know anaerobes, right? If we're creating an anaerobic environment, what are we doing for these guys? We're creating the habitat just for them, right? They're going to be happy. They're going to grow abundantly. If you stuck an aerobe in there, though, and then put these gas packs in there and you generate an anaerobic environment, what are you going to do to these poor aerobes? You're going to suffocate them. You're going to kill them. They're going to die. There's going to be no growth when you open up that jar later if you maintain an anaerobic environment. Now your facultative anaerobes. What's going to happen to them? If you take a facultative anaerobe and you stick it in this anaerobic jar and make this environment anaerobic, they're going to grow, right? Are they going to be as happy as they are in this aerobic environment of our classroom? No, they're going to grow less, but they're still going to grow, right? Because in this case, they're going to ferment because no oxygen for aerobic respiration. What if the environment inside the jar is aerobic? And how would we know if it was aerobic? Our methylene blue, right, tablet or strip would turn blue, right? We would know we sprung a leak, right? Our jar isn't working properly, okay? But our organisms themselves, if we know what they are, would give us some clue, right? Because if you put an anaerobe in there, right, and under aerobic conditions, what's going to happen to it? It's going to die, right? It's going to die. No growth. Your aerobe, on the other hand, which you would expect to die in this jar, would actually be growing quite abundantly. Same thing with your facultative anaerobe. Instead of its growth being slowed down, under aerobic conditions, it's going to be quite abundant, right? So you're going to see all this increased growth. So this experiment, if we were to do it, instructs you to grow some organisms inside the jar. So if we do that properly, what type of environment is that going to be inside the jar? Anaerobic without oxygen. And outside the jar would be aerobic, right? And again, the methylene blue strip would be colorless, right? So what is the environment inside the jar? If we do it correctly, it's anaerobic. Is oxygen present? No. What's the environment outside of our jars right now? It is aerobic. Is there oxygen present? Yes. Otherwise, we'd all be dead, right? So in this experiment, if we utilize Pseudomonas, right, which I'll tell you, it does not grow under anaerobic conditions, but it will grow under aerobic conditions. How would we classify Pseudomonas? Is it an aerobe, an anaerobe? Is it a facultative anaerobe? It's a strict aerobe because, again, growth is only outside in the aerobic environment. Make sense to everybody? All right. So it's our obligate strict aerobe. Clostridium sporogenes, which is another reason why we don't do this experiment, because it's really hard to keep this one happy, because notice, growth inside the jar, no growth outside, right? Where there's oxygen. So this one would be our example of a strict anaerobe, right? Staphylococcus aureus grows inside the jar, but if you compare it to the growth outside the jar, there's more growth. So this would be our example of a facultative anaerobe, right? So you'd really have to be able to compare, right, the amount of growth. So based on the information, Pseudomonas aeruginosa, does this organism require oxygen for its growth? This was our obligate aerobe, so yes. And how does it affect its growth? It actually makes growth possible and allows it to grow abundantly. Clostridium sporogenes was our anaerobe. So does it require oxygen for growth? No. In fact, oxygen would do what? It would kill it. Staphylococcus aureus, does it require oxygen for its growth? Doesn't require it, right? No. But how will oxygen affect its growth? It'll grow abundantly, right? Because it can utilize it. So these are examples of plates put under those two different conditions. So notice that we have aerobic conditions outside the jar, anaerobic inside the jar. And they streak 3 different organisms all on these plates in the same orientation. So number 1, right, represents the same organism under the 2 different conditions, right? So notice that under aerobic conditions and anaerobic conditions, it still grew. And we actually see a difference in the growth, right, under aerobic conditions. It's even a different color, okay? So what classification would this organism be? It's growing under both conditions. This is our facultative anaerobe. This would be our Staphylococcus aureus. Number 2, under aerobic conditions, no growth, right? It was killed. Under anaerobic conditions, do you see that there's growth there? Oh, excuse me, growth, wrong spot, right? So no growth, growth. You see that? So this one would be, number 2 would be, this would be our strict anaerobe, only growing under anaerobic conditions. This would be our Clostridium sporogenes. Number 3, we have quite abundant growth under aerobic conditions, and we have very decreased, probably killed off, right? You see how you can see where they streaked it, but probably these organisms are dead, right? So this would be our aerobe, right? Only growing under aerobic conditions, killed off under anaerobic conditions. So this is the experiment that we did, all right? And again, we didn't have any Clostridium sporogenes because it's really hard to grow anaerobically. What's up? Oh, sure. So here's our Pseudomonas. So you guys have noticed probably last week that it has this green colorization when it grows, right? This one produces a greenish discolorization. So notice there's growth on this plate, there's that colorization. In this plate, there's no growth. And if you read the top plates here, right, were in the anaerobic jar under anaerobic conditions. These plates at the bottom were at outside the jar in our lab under aerobic conditions. So we have growth here, right, for our obligate aerobe. Staphylococcus aureus, we've already said can grow under both conditions, right? And it'll grow better under aerobic conditions. You can kind of see that this plate is almost completely covered as opposed to this one's kind of streaky. Okay, now E. coli, based on the information we've learned so far, what would you say about the classification of this one? It's growing both aerobically and anaerobically, right? Do you see that? So it's probably facultative anaerobe, right? You can grow under both conditions. So as we said, Pseudomonas, right, based on what we saw, right, it requires oxygen for growth, only grew in the presence of oxygen. That was our obligate strict aerobe. Staphylococcus aureus, again, didn't require oxygen to grow. It did grow under anaerobic conditions, but grew also under aerobic conditions. So therefore, it is a facultative anaerobe. Same growth pattern for E. coli as Staphylococcus aureus. So it is also a facultative anaerobe. So an alternative to the anaerobic jar is a candle jar. So you'll see I have some old pickle jars here. You could put a candle in this jar and light it and seal up the jar. What happens is when the candle's burning, it's going to eliminate most of the oxygen, not necessarily all of it, okay? So your really strict anaerobe isn't going to grow. But another gas is produced from the burning of the candle. Actually, a couple of gases, but anyone know which one can be utilized by organisms? CO2, right? Carbon monoxide is also produced, right, when you burn things. That one's damaging, especially to us. But carbon dioxide, and so because of the decrease in the oxygen, microaerophiles would be really happy in this environment. And because of the increased levels of carbon dioxide, our capnophiles would grow. And so these are ones that require higher than normal levels of carbon dioxide. So you'd either have to pump in carbon dioxide into an incubator or grow them in this particular environment in order for them to grow. So anaerobic jar, right? Why am I doing this experiment? How is it performed properly? What outcomes? And what does this data mean? So can a particular bacterial species grow in the absence of oxygen, right? Less than 1% atmospheric oxygen is the question we're asking when we perform that experiment. Right? Again, we've got to put organisms in. We have to put the gas pack generator in it. We have to actually generate that anaerobic environment in order for the experiment to work. And then we look at the different growth patterns, right, to determine their ability. So FTM tubes are going to give us that information for our organisms, right? We're not obviously, as I said, doing and setting up these jars because we don't have enough for everybody to use. Our last one that we're going to talk about and that you guys inoculated last week is our motility test. And this is just one of the tests that we'll do. You'll read about another one for homework that we'll do on our organisms next week as kind of a confirmation. One of the problems with this test are some things that can go wrong, right? And so more than one test will give us more accurate information. So is the media used for this test solid? And the answer is no. It's what we call semi-solid. So if you were to touch it in comparison to what we utilize for plates, what we pour into plates, or even those slants, this one would be squishier, right? Because there's less agar, less of that solidifying agent in that media. And we also use an indicator for this test. And it is TTC is the abbreviation for it. And it's colorless, although we've been having trouble with that. So our tubes have come out a little pink because it's been getting reduced. And so when it reduces, we get the red color, right? So if we didn't have that indicator in the tube, sometimes it would be really hard to determine if the organism moves. So remember how you perform this test is you stabbed right in your organism. So you would have inoculated hopefully along a nice straight line, And so if the organisms are non-modal, they will stay exactly where you put them. The reason why it's semi-solid is to give them some space between the molecules to move if they are modal, right? And so they would diffuse out. So you notice how this one's kind of cloudy, but it's kind of hard to see, right? That these organisms moved. Where in these bottom ones, where it has the indicator, you can clearly see this organism stayed right where we put it, right? And this reagent is insoluble, right? It does not move on its own when it turns red, when we reduce it, right, by the growth of these organisms. So you'll see this nice straight line. If yours is modal, it's going to spread out. In the case of our tubes, most of them too are going to look like it just completely engulfed the tube, right? That they've spread all throughout the tube if yours is modal. Sometimes though, you'll see kind of this tornado effect, right? But because of some of the trouble we've been having with the media, we don't have any of those pretty ones for your guys' unknowns. I do have examples in our rack that I'm going to go through with you guys, right, to review before you look at your organisms. So as I said, on page 6 of your lab manual, it uses some of the terminology and some consequences that can happen for some of our tests. So a false positive is where you observe results, right, that look positive, right? So it may look like it's spread all throughout the media in this example. But in fact, your organism is non-modal. So this would be a false result. How would you know you got a false result? Right? We run controls, right? And that's why I have ones for you. And that's why sometimes we'll run multiple tests and why we have to take into consideration how the tester performs in the particular things that could potentially go wrong. Okay? So false negative would be, it looks negative, right? But is the organism actually negative, in this case for motility? No, right? So what are some things that could have gone wrong, right? So these are the things that we'll talk about. What could give us the possibility of it looking negative, but the organism is in fact positive from its motility? Some things that come into play for our tests is their sensitivity and specificity. So sensitivity is the limit of the test, right? The range in which we can discern false negative from true positive results. And probably the easiest one for most people to understand is pregnancy tests, right? Pregnancy tests have a certain level of sensitivity, right? They tell you you can only test after so many days after your missed period, because that test has a certain range that it can detect the hormone present, right? It has to be at a certain level for that test to detect. So as you increase longer in your pregnancy, that level, that hormone goes up, right? So once you've reached a certain point, right, you may be within that detectable range, okay? So a lot of tests, right, have a sensitivity range where you can actually see positive results, okay, and not end up with false negatives. Because even though it may be there, it may not be detectable. You can't see it, right? Or the test cannot let you know that it's there. Does that make sense to you guys? Where specificity is just that. How specific is this test, right? Can it come out positive from something else? So again, I'll bring you to the pregnancy test. So I don't know who thought of this, but somebody thought of testing male urine with a pregnancy test. That should definitely not come out positive, right? Last I checked, guys do not get pregnant, right? But guess what? There is actually a chemical that's produced when males have testicular cancer that can be detected by the pregnancy test. So it's similar, right, to it. So the specificity of this test, although really, pretty specific for that particular hormone, for women, it's detecting something else in men, right? what I don't know, right? That's a recent finding, and how or why, like, they decided to do that. All I can think of is like, well, honey, since you've got a PLP2, and we'll test mine just in case, But most of the tests that we'll run in this lab are pretty specific, but there are some that we'll talk about where they have limits in their specificity, right? Where depending on how you perform the test, right, you may be detecting the presence of something else than what you think you're detecting. Okay? Make sense? Okay. So mostly we'll be dealing with sensitivity of our tests, but remember, we also have to know, what are we testing for, right? And is this test truly testing for what we're asking? Are we having some cross-reactivity like what happens with the pregnancy test? So to help us in these, we'll usually run controls, right? Known positives, known negatives, so that we know and can see, right? And most of our tests are going to be yes or no, right? Is it positive? Is it not, right? But some of them can be quantitative, right? And in that case, you would run a series of controls at different concentrations, right? Quantitative means that we can assign a number to it, instead of being qualitative, which is just yes or no, right? And so we refer to those as standards, right? When you're going to do controls, right, where you can actually measure it out. And again, we're not going to run any tests that we're going to do different levels, okay, where we'll actually get a number. We're going to be dealing just qualitative yes or no. But one of the tests we will run later in the semester can be quantitative, where we actually get numbers, but we haven't worked it out to do that. So we'll compare our unknowns, right, what you don't know, to our controls to help us in determining whether we have positive or negative results. But if you had where you could do a standard, right, you would have a standard set of controls at different concentrations. You could plot this out, and then based on where your unknown falls, you could figure out the concentration of your unknown. And so a lot of scientists do this for protein concentrations, for antibody concentrations, for antigen concentrations, when we run some of the different tests. And so our unknown sample, right, you can do a linear regression, or you could do one that follows the actual dots, which will give you a more accurate reading. So mathematically, when you use the curved, you actually end up with the other side as well. Obviously, we use the side that would fall within the range. So your book, unfortunately, has a potential typo, right, when it comes to talking about false positive and false negative results. Assuming we haven't made any huge mistakes, right? False negatives indicate usually a sensitivity issue with your assay. False positives usually indicate a specificity problem, right? You may not be testing truly for what you think you're testing for. So there's a mathematical way of calculating. Again, we're not going to do this.
That's where the error occurred in the book, however. They had
Yeah.
to perform this.
Yeah. Which one is wrong? One of these,
They
the
used
bottom
same
one.
denominator for both equations, but it's bad.
Oh, yeah, it should be different. So for the motility test, why is it important to insert, right, and remove the needle along the same stab line? So here's some pictures of why. These are bad stabs, right? And some of you guys may have some not so great stabs. And again, it just comes from practice of the technique. So this particular tube, when you turn it this way, right, you see how it's pretty flat, but then you turn it 90 degrees and you see how it's kind of spread out. What happened here is the person went in and then came back out at an angle, right? So they spread the organism as they were inoculating it. So is this positive for motility? No, right? If it was positive for motility, if you were to turn this tube, you would see spreading all throughout the tube, right? All throughout the tube, right? You turn it 360 degrees. So you'll notice when you guys are recording your results, we want you to record two angles, right? One 90 degree or 180 degrees from each other or whatever. So if you have one of these flat planes, right, or a spread plane, you need to indicate that. But remember that this is an inoculation problem, right? So don't record this as positive, right? Because if you did, it would be a false positive, right? Same thing here. Notice they stabbed it twice. Didn't realize they already stabbed it, right? One stab here, one stab here. Okay? This organism didn't move. Right? So growth will occur anywhere that you inoculate, right? Since spreading from the stab line indicates motility, right? If you do a poor stab, right, and you don't notice that, you may think that it's positive for motility. So a single stab is much easier to, of course, interpret, right? But not necessarily the easiest thing to do. So what are some possible ways you might get a false positive and false negative result? So we already said with false positive, right, it would be you have an organism that's non-motile, but the results look positive. And this really has to do with how we perform the test. So we've already said, what could you possibly do wrong where it might look positive to you? A bad stab, right? A poor stab, right? So, and in this case, right, here's a nice clean stab. Here's one, it's a pretty clean stab, right? But what happened is that there were pockets of openings in the media where the bacteria were able to move through, right? So you see how it's uneven? So this again is a non-motile organism. So this is a problem with the media, right? Not necessarily the person's stab. So it's another thing that could go wrong, right? Is the media may allow it to look somewhat diffused. And again, anywhere the organisms are, it's going to turn red. So what about negative, right? So here's an example, right, of what might be a false negative. This is where it looks negative, right? You think it looks negative? Can anyone give me an indication on why I'm like, well, I don't know if it's actually negative. Didn't stab it properly out. What are you noticing about this line? Nah, straightness doesn't matter. Look at the actual line. It's scarce, right? There's very little growth, right? So did they inoculate enough organism? Did this organism maybe not grow in this type of media, right? We know certain organisms need certain types of food. Is it maybe not growing, right? This is one that you would definitely test again because there's such little growth, right? You would test it again, or you would look at it under the microscope like we're going to do next week. Because this isn't a very strong growth. So not inoculated with enough live organism might have been the case for that one, and that would give you maybe a possible false negative result. The media might be too solid, no worries. The prep person does know how to make the media, though we are having a little bit of difficulty with it right now. However, as the media ages, it will dry out as well, right? And so that would affect, right, how well they move through the media, right, and some of those pocket problems that we sometimes see. So Why is it essential that the reduced TTC be insoluble? What does insoluble mean? It does not, it's not soluble, it doesn't go into solution, right? So it stays right where it is, where it's been reduced, right? So what does soluble or insoluble mean, right? Soluble means it diffuses through a liquid media. And remember, this is semi-solid, so there's a liquid component to it. Insoluble, right, is when it's reduced, and so when the organism reduces it should stay right there next to the organism. So we're only helping us visualize where they're growing. And so it's colorless, right? It spreads all throughout the media. But again, we're having a little bit of difficulty with it. So we have a little bit of it changing outside that zone. So what's the question we're asking, right? How do we perform the experiment properly? What outcomes should we record? What does this data mean? So is the organism motile, right? Can it move on its own, or is it non-motile, right? Remember, we have to properly stab it. The TTC is going to help us in seeing where the organisms are growing by the fact that it reduces, becomes insoluble, stays put, and it is red. So what are we going to record? Right, we're going to record growth along that stab line. We're going to even okay. I'm trying to just, anyways. Okay. If it is motile, we should see growth throughout, right? Or we might even see that little tornado effect. But again, if you turn your tube 360 degrees, you should see uniform growth out from your stab line, right? That would indicate a positive result, right, for an organism being motile. Flagella are the most common way in which bacteria are motile, but it's not the only way that they're motile. But I can say for a fact that the ones that we're utilizing in this lab have flagella. That's how they're motile. We don't have any spirochetes. Remember, those can corkscrew through. We're not working with any of those. Some organisms can produce slime and kind of glide along solid surfaces. Others can kind of twitch and move. But all the ones that we're dealing with are movement by flagella. Okay? So they're swimming through, being propelled by their flagella. So let me go ahead and end this.